Okay, everybody. In the interest of staying on time, we will get started. So I am Tommy Sutor. I am an investigator at the Bronx Veterans Medical Research Foundation at the James J. Peter VA Medical Center and an assistant professor at Icahn School of Medicine at Mount Sinai. And today our webinar topic is N-of-1 study designs for spinal stimulation studies. And we have two fantastic speakers with us today that are going to be telling you about the pros and cons of this type of study design from both a research and clinical perspective. So you can put any questions that you have throughout the webinar in the chat, and we will be monitoring those, and we'll have some time for Q&A at the end of the webinar. So our first speaker is going to be Noam Harel. He is a professor of neurology and rehabilitation and human performance at Icahn School of Medicine at Mount Sinai, and he's also a staff physician at the James J. Peters VA Medical Center. His primary interests are improving motor outcomes in spinal cord injury and ALS, and his clinical research lab uses targeted physical exercises, electrical stimulation, medication repurposing, ischemic condition, and other methods to reactivate weakened nerve circuits. So with that, Dr. Harel, please take it away.
Thank you, Tommy. Thank you, Jake. It's great to be here. Dr. Weir and I collaborate and have collaborated for years on a variety of projects. So we're going to talk about N-of-1 studies design today. And I think some of the problems, just to recap in our field, are obvious to all of us. Just want to recap as part of the setup to this talk. Okay, we have small sample sizes. We're not heart attack. We're not even stroke. Okay, spinal cord injury studies are plagued by small sample sizes, which makes studies harder to interpret and contribute to this whole crisis of irreproducibility that we have. Okay, so that's one obvious major problem in our field. Another problem in spinal cord injury research is every individual is different. Okay, every spinal cord injury is a little bit different, especially when you take into account people's associated conditions, their secondary consequences of spinal cord injury. Everyone's going to need individualized treatment. And pills are probably going to be part of that treatment plan, but a lot of what we do in our field, whether it's different types of exercises or stimulation, they're not standardized. They're either impossible or difficult to blind. And it's just not like taking a pill. That makes it harder for our studies to get standardized, reproducible results. And I think we can all agree that we're probably going to need to really move the needle, clinically move the needle for spinal cord injury. You need multiple modes of treatment combined. Okay. Exercise is definitely going to be one of those treatments. We may need one or more drugs. We'll probably need some form of stimulation. We may need stem cells, et cetera, et cetera. There's multiple modes of treatment. Some combination is going to be necessary, but this is the quandary. We all have thought about this, and it's a difficult question. How do we test each component of that multimodal treatment for safety and efficacy? Okay. For example, if you want to combine these three modalities, you'd have to have a placebo for exercise, some sort of placebo for the drugs, and some sort of placebo or placebos for different types of stimulation. Okay. And again, this is just meant to show for spinal cord stimulation, there are numerous factors to test, and some of them require more than one control. Okay, how do we test all of these at once? Imagine if this was a randomized controlled trial, a parallel randomized controlled trial. Imagine how many groups you would need in that trial to test all of these in one study. Or imagine you're only going to test one thing at a time in each RCT. Imagine how many years that would take. Okay, so this is a quandary we have. Okay, so there's no magic answer to this, and we're not trying to oversell N-of-1 design, but what if there is a way you could actually have a valid approach to testing individualized treatment, multiple types of treatment within the same study in a small sample size? And yes, that's what we're going to talk about today. It's N-of-1. It's not the panacea for everything, but We're going to try to point out some of its advantages as well as the disadvantages. Okay. Um, so N-of-1 study design is also called single case experimental design. I think both of those names are bad names. We need a different name for this type of study design because inevitably, oops, people will assume it's an N-of-1, which it's not. And people will say this is the same thing as a case series, which it is not. Okay. We need better design. Hopefully, someone out there in the audience can start suggesting a better name. Sorry, not better design, but better name for this type of design. Okay. So, what is an N-of-1 design? The example down here is from a GI study. But for each participant in a trial, you randomize that participant into one or more treatment arms. You have them undergo the treatment, you measure the outcome. Then you wash out, in this case, it's drugs. You wash out the drugs, randomize them again between the two drugs, measure the outcome, and so on and so on. You're doing multiple crossovers in each participant. These can be blinded, and you're measuring the outcomes after each phase of the study. That way, from each participant, you get more data with themselves as the control. And then you do this again and again on multiple participants. It's not really an N-of-1. And it's not a case series in terms of we're not just following the natural history of this patient's treatment. This is a prospective type of study design. Okay. So what are the ideal conditions for N-of-1 study design? Soon to be renamed N-of-1 study design, I hope. You need something that's relatively stable, a condition that's stable, because if there's too much, lability in the condition or the condition steadily getting better or steadily getting worse, it's just going to make it much harder to measure or discern the effect that your intervention is having versus the natural history of the condition. So you need a stable condition. It's really useful when a condition requires individualized treatment and multimodal treatment. And it's also really helpful when you're having trouble getting larger randomized control trials funded. Okay. So can we, you know, makes you go, can anyone think of a condition that might fit these bullet points? Right. So I think chronic spinal cord injury is an obvious place where we can apply this type of design. Okay. By the way, we're here to try to sell people on this design. It's not a cure for everything, but probably more of you out there are already familiar with the design than you realized. Okay. These are some very important papers published in the field of spinal cord stimulation. Many of you are probably familiar with many of these papers, okay, from the biggest groups out there in transcutaneous and epidural spinal cord stimulation. These are all done in 6 or fewer. Most of these were done in significantly fewer than 6 participants. Okay. Each participant is studied extensively, and that takes a long period of time, but they were able to go through multiple stages, parameter twiddling, tweaking, and parameter optimization over a period of time. And, Reggie Edgerton passed away recently. One of the times he spoke after the talk, he made a comment that has stuck with me. He said, "We don't study a lot of patients. We only study a few patients, and we study the shit out of them." That's what he said, and that had an effect on me for sure. Okay, I think he was pretty successful. All right. So, what are the advantages of this design? Okay, because you're not doing Group A versus Group B, there's less of an emphasis on homogeneous groups, so you can have broader inclusion criteria. It's individualized, as we discussed. And depending on how long you have and how long it takes to do washouts, you can test multiple things, multiple interventions, or even combinations within one person in one study. Okay. And because you're tweaking for a long time with each person, that participant has the opportunity, hopefully, to directly benefit from the study. And that contrasts with a lot of earlier stage RCTs where you're doing Group A versus Group B. and it's only an early stage study, that participant is contributing to the field, but they may not have a chance to directly benefit. And especially as Professor Weir will show, you can get more data out of each person than you can in a regular RCT. And we can get useful information even with a small n. And again, those papers on the prior slide were all major papers that have impacted our field. Okay. Again, this is not a perfect solution to everything. There's some trade-offs, some challenges. Okay. Definitely going to do fewer participants with the trade-off being you can test more parameters in each participant. Okay, but it is a disadvantage. Not every person with spinal cord injury or any other condition, not every person has 6 months, 9 months, a year to spend coming in and out of the lab or your center over and over again. So participation takes a lot longer, and you have to be with any crossover study, whether it's an N-of-1 design or other crossover designs, you have to worry about washout, which is a major issue in all crossover studies. And you, let me just say, larger N is always better, including an N-of-1 design. If you study 20 people in an N-of-1 design, you will more likely get generalizable information. Okay. If you study 3 people in N-of-1 design, you may still get generalizable information, but it's a higher odds of not being quite as generalizable. Okay. So we'll talk more about that and the statistical challenges involved. Okay, so this is just an example of one of the studies where we're taking this approach. We have an ongoing study of paired stimulation aimed at the cervical spinal cord to try to, strengthen synapses in the cervical spinal cord that are going out to hand muscles. So the population is people with chronic cervical spinal cord injury. Again, the intervention is paired or synchronized brain and spinal cord stimulation, etc. So there are a lot of variables that need to be tested just for this one intervention. When you're talking about stimulation, there are a lot of stimulation variables. It's not just flicking a light switch. We have to test a lot of these things. We don't know enough as a field how much each individual responds to different parameter combinations in these types of studies. I'm sure many of you out there are used to 30 hertz as a frequency of spinal cord stimulation. There have been studies looking at a range of frequencies, and really some earlier studies kind of found anywhere between 15 and 60 hertz might be a sweet spot. But since then, as far as I can see, everyone just sticks with 30 hertz because it's a field standard, but that's a little bit arbitrary. Okay, so we want to tweak things a little more within each person. And this is just an example of the data, and it's not simple. Okay, N-of-1 doesn't mean it's simple. It's actually complicated. This person is a 62-year-old participant. She has incomplete spinal cord injury, cervical spinal cord injury. She's done 48 sessions with us so far of different variations of the paired stimulation with or without exercise, hand functional task practice exercises combined in different ways. There are multiple repeats of each intervention. And this is just a raw picture of the data on how well her target hand muscle responds to brain stimulation or motor evoked potential after the treatment versus before. Okay, so many of them are positive. Is any one dominant or over any of the others so far? I would say no. And this hasn't even been analyzed uh, um, in terms of statistical testing yet, but Joe Weir will discuss that. So it's just an example of how you would start looking at it. This is all data from one person. So you can see how this is generally more data to analyze per person than in an RCT. We have another study that Tommy and Jake have been and are a part of, where spinal cord stimulation is being used to optimize blood pressure stability in people with chronic cervical SCI. Not sure whether that's happening. And again, anytime you do spinal cord stimulation, there are a lot of factors that need to be tested. So they've taken this similar approach to which level, which intensity, which frequency of spinal cord stimulation has various effects on the systolic blood pressure. And again, Professor Weir is going to show this diagram in slightly more detail in a little bit. Okay, where are we? Okay, actually, when I said a little bit, I mean pretty much right now. I'm going to pause here, and Joe Weir is going to talk more about the statistical challenges and approaches to analyzing these results. So I'm going to stop sharing. Hope this works. And Joe, ready to go?
And yeah, thank you, Noam. That was great. And I'll just quickly introduce. So, as Dr. Harel stated, Joe Weir is a professor in the Department of Health, Sport, and Exercise Sciences at the School of Education and Human Sciences at the University of Kansas. And his research interests center on statistical analyses, neuromuscular physiology, and the autonomic nervous system. Much of it centered on the application of multi-level modeling to physiological and biomechanical data with particular interest in spinal cord injury. So, Dr. Weir, if you would like to please continue, take it away.
Thanks, Tommy. Can you see the slides now that I
It's
shared? Okay.
Go into
in
presentation mode.
PowerPoint, not
Yeah.
yeah, there we go.
Okay. So make sure I'm getting on the right slide here. And thanks, Noam. Um, so some of the issues that come up, let me preface this by saying I kind of got pulled into this N-of-1 stuff a little bit kicking and screaming. So I'm a traditional um, researcher, exercise physiologist, and I like randomizing people to different conditions and repeated measures designs and all that type of stuff. But as we started uh, working on these types of spinal cord stimulation studies, both in the neuromuscular realm that Noam is working in, looking at motor evoked potentials, and then with Jill Wecht and Tommy and Jacob looking at blood pressure normalization. Um, all of the stimulus parameters that are available leads to so many combinations. You really can't design a clean study in a traditional sense to look at a lot of these types of questions. So some of the things that we sort of have to think about when we're analyzing this data, and there can be a lot of data uh, from one subject, hundreds, if not thousands of scores. So there are order effects that potentially are an issue both within a participant that may influence how the data look, and also if you have multiple subjects that are sort of we're pooling N-of-1 types of analyses, those order effects may affect how different people are responding to these different sort of interventions. Independent. So when you remember back to your stats classes, they would talk about linear models and use the abbreviation IID. So one of those I's is independence of scores. So um, when we have repeated measures data, like you will in a single case type of study like this, these scores are not independent. That is, a person's score in so their TMS value, excuse me, their motor evoked potential amplitude. If you're doing three or four of those MEPs in a row with the same stimulus parameters, you can sort of estimate what the next value will be based upon the previous value. They're correlated with each other. So that does affect how we think about how to analyze the data. And the order of scores. So there's a temporal sequencing that will be happening if you're getting multiple scores within a given stimulus setting. So scores that are closer in time are going to be more correlated with each other than scores that are further separated in time. All that means that there are kind of some SCED specific calculations out there that, frankly, I'm just starting to become familiar with. So a lot of the standard effect size measurements, like a Cohen's D, weren't really set up for these types of calculations. And on a subsequent slide, I'll have a reference to an R package, for example, that calculates effect sizes that are specific to some of these single case study types of designs. Okay, this is typical picture for a classical ABA single subject design. And lot of my friends and colleagues in Ed Psych, for example, on the measurement side, have been doing this type of work for a long time. So in single case or single subject design studies, one of the key features is that you're getting multiple scores from each person so that you can establish what looks like a reasonable baseline. So here, the A condition in the top panel um, shows a bit of an upward trend, but on average, the scores are higher than they are, whoops, in the B condition and so forth. And then when you go back to the A condition, they trend back up again. So there's a tradition, as I understand it, in the N-of-1 world where a lot of the analysis is basically plotting the data and looking at it. And in some cases, there's really not any statistical analyses done. But you're just looking visually that, yeah, if you could kind of ballpark what the trend is here in A versus B and then back to A again, you see something different than you see in the bottom panel where A and B all look to be rather similar to each other. So step one, whether it's single subject design research or a more traditional type of approach, is always plot the data, always visualize the data first before you even start calculating means and standard deviations. And that certainly is important, the important first step for single subject design. Okay, so here's the image that Noam had earlier, and we stole this from Tommy. And so we'll blame him if you don't like the graph, but we both stole it independently, so I guess we both like the graph. So in many ways, this is a three-dimensional graph, right? So our dependent variable is systolic blood pressure on the left-hand y-axis, and then stimulation amplitude is the black line, and on the right-hand y-axis. And then we also have time here. So this protocol was administered in a sequential fashion. So I'm not sure why the slides are automatically moving forward, but the stimulus amplitude is progressively increased over time. And once again, step one is to plot the data and look at it. So we don't have a nice necessarily linear trend, right? So as the stimulus amplitude increases, the blood pressure increases up to a certain stimulus intensity, and beyond that stimulus intensity, we're not getting further increases in blood pressure. And to further complicate this, the goal is not to make the blood pressure as high as we can. The goal is to normalize the blood pressure. And what, Tommy can correct me, but I think we're trying to center it around 120 millimeters of mercury with maybe a plus or minus 10 millimeter mercury range around that. So some of our analyses actually are binary. Is a given pressure in the optimal range or is it outside the optimal range? And then you're looking at some sort of variant of a logistic regression type of approach. One other point I want to make here is that generally speaking, even though we're showing the means at each stimulus intensity, we're treating each of these pressures as a discrete score. So we're not throwing away this data. And in fact, there may be some analyses where we're more interested in the variability that we're seeing than in what the actual pressure values are. Okay, but I do want to talk about clustering here for a minute, or nesting. I kind of use those terms interchangeably. There may be some differences, but the idea here is that here's maybe a more traditional RCT type of design. So you got a multi-center trial. So we got, in this simplified example, two different hospitals. And within each hospital, you've got different physicians who are administering the treatments. And then within each physician, you've got a variety of patients that they're treating as part of this RCT. So clustering has to be accounted for in these types of designs because these scores are not independent. that patients who are treated by Doctor 1 in Hospital A share some correlation with all the other patients that are treated by that same doctor at that hospital. It may be socioeconomic characteristics, sex differences, what have you. But there is some sort of correlation between these scores that we need to account for in our statistical models. Well, the same thing applies to even a single subject design. It potentially applies, not necessarily. So here we have one subject, and we'll simplify this a little bit. So we're looking at two different stimulation sites for this particular subject. And within each stimulation site, we've got two different waveforms. So it could be monophasic versus biphasic, let's say. And this stimulation site might be a lumbar region. This might be a thoracic stimulation site. And then within each of these categories, we will have multiple stimulus intensities. And within each stimulus intensity, you're going to have multiple scores. So here it's systolic blood pressure, but with Noam's data, it might be motor evoked potentials. So certainly with blood pressure, right, you can get a lot of pressure values that are all nested or clustered within each stimulus intensity, which is then nested within the waveform and so forth. So I'm going to talk, use at least a couple slides here, talk about multi-level modeling in this type of context. So multi-level modeling sometimes is known as mixed modeling or hierarchical linear modeling. In some ways, it's just an extension of standard regression analysis. So you're going to get slope coefficients and confidence intervals and so forth, but they're a little bit more complicated to pull off and they're not quite as intuitive. But we employ these multi-level models because we have this nesting. We have this lack of independence of scores. And once again, not all single study designs are going to need multi-level modeling. In fact, most of them probably won't. But for the types of stuff that we're doing, there is this clustering that we need to be paying attention to. So we've got repeated measures, so multiple scores from each participant. And that's just true for all the types of research that we do. I have a hard time coming up with examples where we have only between subjects types of designs. We include repeated measures in almost everything. Back when I was going to school and up until I started using multi-level modeling, we would try and force all of our repeated measures designs into a more traditional repeated measures framework or a paired t-test framework, which still works depending on the situation. But multi-level modeling gives us a lot more flexibility than forcing everything into a traditional repeated measures framework. And it allows us to do analyses like we're going to be talking about here. Okay, so just some terminology with multi-level modeling or mixed modeling. So when people refer to these types of models as mixed models, they're mixed because they contain both what are called fixed effects and random effects. And it's not always intuitively obvious which one is which. In a traditional study with repeated measures, subjects are random effect because we're not interested in each subject per se. We've taken a random sample of subjects and are studying them to try and make generalizations to the population at large. And so typically, I think hardcore statisticians might not appreciate me characterizing it this way, but typically fixed effects are typically the variables that we're interested in quantifying and testing for statistical significance if we want to do that, or calculating confidence intervals and so forth. Okay. I'll skip some of the other details here, but another way of thinking about fixed effects is the effect is fixed because we're conceptualizing it as being a constant or average effect across all the clusters that we're trying to quantify. So it may vary on an individual basis, but the overall or average effect of some variable like stimulus intensity or like monophasic versus biphasic, that type of thing. And one of the other advantages of multi-level modeling in a traditional sense is that we can deal with missing data. So we don't have to use listwise deletion or data imputation. For the types of studies that we're talking about here, it's not missing data per se, but it gives us a lot of flexibility in terms of we don't have to have the exact same stimulus intensity across different parameter settings. We don't have to have the same number of motor evoked potentials. In a strict study might be, well, we're going to do 5 MEPs. Well, maybe we'll do 6 MEPs and under one condition and 8 in another. And these types of statistical models can handle that without a whole lot of difficulty. And just to give you an example, and Noam talked about this already, if you look at all these stimulus options that are possible, so with the blood pressure data, and this might not even be complete, Tommy can correct me. So there was at least 4 different stimulation sites that people were looking at. There was monophasic, biphasic. There was also a pseudo-monophasic. That you look at the possible combinations here, that starts to be kind of a big number, different pulse width settings and different frequencies of stimulation. And we're going to talk about carrier frequency, but different types of waveforms, was the carrier frequency present or not. So that adds all this level of complexity. And as it turns out, we are not trying all these possible combinations with each person. So there's this adaptive protocol that when the participant comes in, as I understand what's happening in the data collection, certain settings were tried, but based upon the response of the participant, that led to the next decision in terms of what the stimulus parameter was that was being assessed. So if you're trying to do this in a traditional experimental model, you've got a whole lot of different cells here, and you'd have to simplify your study, as Noam pointed out, because you couldn't look at all these possible combinations. And in fact, we're not with the single case study. We're not applying these to all the participants. Okay. I don't, I don't know if this is take home or parting shots is maybe probably how I should have put this. Despite the fact that I spent a fair amount of time talking about multi-level modeling and using multi-level models, not all N-of-1 designs are going to need multi-level models. And especially if you average across a bunch of blood pressures or a bunch of motor evoked potentials. In some cases, you sort of wash out some of that nesting. I don't know that that's a good thing, actually, because leaving the individual scores as individual scores gives us some advantages, not the least of which is improving some of our statistical precision. But even if you have a design that doesn't require all of these possible permutations, like with the spinal cord stimulation, there are single case experimental design specific effect size calculations. So as I pointed out earlier, just a regular old Cohen's D is probably not going to be the approach you want to take for looking at the effect size of an intervention in a single case design. And for those of you who use R, there's a package out there, single cases ES, so single case effect size package. You can just Google this. There's a GitHub page where you can download the package. To be fair, to be honest, I've not actually played with that yet. But there's a variety of approaches in the single subject design world to quantify effect sizes that are a little bit different than what we're traditionally used to with more traditional experimental designs. And I'll emphasize this again. Look at your data. First step, always look at your data. And that's going to give you as much, if not more, I think, insight into what's going on than a lot of the fancy statistical stuff that we like to play with. All right. And I think from my end, that's probably the end of the slides I was going to address.
All right. Well, thank you both so much for those fantastic talks. For those of you who might not have seen during their talks, I did remind everybody that we'll be taking questions via the chat. So if you have questions for either one of our speakers, you can go ahead and put them in the chat. We already have a few to start, and it it actually looks like Noam is on top of answering some questions via text in the question and answer box. So I'll start with one over in the chat box. So one of our attendees is asking, and this is a broad one, it looks like for either speaker. In N-of-1 designs, it was mentioned that visualizing the data is the first step in the analysis, which makes sense given the individualized nature of these studies. However, could this also introduce bias in how we interpret the data or choose subsequent statistical analyses? For example, once we observe an apparent trend visually, there might be a tendency to select analyses or supporting evidence that confirm our patterns. So, how can we ensure that interpretation remains valid and isn't overly driven by confirmation bias? Nice, simple question to start off the Q&A
Yeah,
for our
actually, it's a
speakers.
great question, right? Philosophically, I kind of approach this as like the whole reason we do statistics is so that we can't just sort of look at the data and interpret it how we wanted to interpret it from the beginning, right? So I think some of these um, issues are we do have an a priori analysis plan, right? So when we think about the blood pressure response, we know it's not going to be linear. So a priori, you might think about putting in like a quadratic component or something like that. And then hopefully our visual inspection will support that. But if if the quadratic component doesn't help, it's not going to um, improve our model fit. So both the statistical test as well as the visualization hopefully will be complementary there. But I think that's a fair concern, right? But just like any study, as many of these decisions that we can make a priori, the better.
Yeah, and I'll add, we certainly have a priori hypotheses for our paired stimulation N-of-1 design study. Um, but there is a fair amount of changing course depending on what a particular participant is responding, what we think they're responding well to. We might further test a specific parameter for different variations, etc. And I will just also ask a hard question back. Sometimes it works the exact opposite way. We tend to favor our a priori hypothesis and filter everything through that a priori glasses, a priori shaded glasses. So it can work either way, to either go with the subjective or objective data as we go or have an a priori hypothesis. Um, there are tons of great questions. I wanted to follow up on a couple of them, Tommy, that I started answering, but I'm losing them.
If you'd want to answer them verbally for the rest of the audience.
There was one that I said I would answer verbally, and I think I need Joe's help. Yeah, here we go. So,
Okay, yeah, go ahead and ask that one then.
um, well, one of them was answer. Okay, so washout period for TSS. Yeah, unlike medications, we don't know the exact half-life of TSS, spinal cord stimulation, as we do with various drugs. I'd say that's roughly arbitrary. We tend to try to separate sessions by a week in most cases. It probably doesn't need to be a week. It depends how long you're applying the intervention for. So it's a very complicated question, just like everything in science. The more questions you ask, the bigger of a rabbit hole it becomes. So that is a tough question, but I would say the way we approach it, our interventions last on the order of 5 to 10 minutes, actually, only. So we even think 24 hours is probably enough for a washout. And then Linda's question, I was going to ask Professor Weir for help on this one. She's asking how AI can affect our decisions in this design, I guess, in real time as a participant's undergoing a study. I think it certainly can help. but that's not really the answer I think you're looking for. I don't know if you want to try to provide any more details on that.
I don't know. I I cheat with AI all the time now, in helping me to code. But boy, having a um, like a bot that would you could train to help you. I'm serious, right? Uh,
Or
to
give you
make
thoughts.
decisions. I don't know. Would there be value add to that? I don't know. That would be interesting.
Right, we have a oh, sorry, were you done, Joe?
Yeah, I think I was going to give an anecdote, but it's probably not helpful.
Yeah, Tom, if you can drive moving forward, because there are a lot. This is great to get a lot of things, but I'm
Yeah, okay.
going
I'm
between chat
hearing
and Q&A. It's
Yeah, so I'm
confusing.
hearing from one of our behind the scenes panelists that we have somebody who is unable to type right now, so they're requesting to ask their question verbally. So
Sure.
I believe you've been unmuted and you can go ahead.
Hello, can you hear me?
Yep.
Yes.
Yeah,
Professor
this is Keith
Tansey.
Tansey. I apologize for being on the road, but the questions I have, well, there was a couple comments. One was that this has been brought up, this idea that you know that there's we're assuming we can have washout, yet we're in a field where we're trying to change the nervous system.
Yeah.
So we kind of hope that there couldn't be a washout. That we would change the nervous system for the better. So for some of these things, I don't know if this design will help sort out treatment A versus B versus C versus D because it mattered the order. The second point I wanted to make is that the effect is not just which treatment or which parameters, but even things like duration. So it's the effect of the 30th step, the same as the 300th step, it's the same as the 3000th step, not only in the effect it's having then, but the extent to which it may be changing the nervous system. And I just wanted to point out that there are, even if you constrain the parameters to just one thing like frequency in spinal cord stimulation, with 9 patients, we were able to show 4 distinct groups of both the motor
Yeah.
responses and the variability to the frequency. And what was very clear was we did not know what the necessary and sufficient conditions underlying it was. So if you have hypertension or you have a urinary tract infection, you know what the bacteria is, or what the blood pressure is, but we don't know what's necessary and sufficient to get these responses in one individual versus another. So it's not only the small n, it's this heterogeneity that we don't even understand. And then finally, I just want to comment on this idea of a bot or using AI in this. I think there's probably a really interesting possibility of setting a standard of where am I trying to get this person, whether it's normalizing EMG or normalizing forces or normalizing electrophysiological signals. And any intervention that moves you away from that normal profile, you stop. And anything that moves you towards it, you try more of it, or you explore the parameter space around that. So you could probably not try everything and everybody in all possible combinations, but have a decision tree where you would slowly peel back the things that are not really impacting the situation to be able to focus more on the ones that are. And I'll sign over to you guys for comment. Thanks.
And I think regarding the last point, and appreciate the comments, that's kind of what is happening now with these protocols, right, Tommy? But it's a human making these
Yeah.
decisions
We're slow.
as opposed to automating it. One could certainly envision, like if you had wearable stimulators, right? Um, where like to normalize blood pressure, that you could have an AI system that is trained to adjust these parameters that is tuned to that person. Um, and as they're in a free living environment, adjust the stimulation if they become hypotensive or hypertensive, right? That seems like a reasonable possibility.
Yeah, and I may be not up to date on this, but in diabetes, insulin pumps have been discussed forever and been around for a long time. I do not believe, I could be wrong, but I do not believe even with that one parameter, blood glucose, I do not believe AI is quite there yet in controlling it, but it will. And it probably already is there, and I'm just not up to date on that. And I just wanted to comment on Professor Tanzi's first point. He pointed something out that's a philosophical and ethical problem, I think, that we should always keep in mind. We're talking about things like washout, and you're absolutely right. That goes against the person's interest to wash things out. What if it had a good effect, and then we want to wash it out for scientific reasons? There's always a catch, right? So we have to weigh these what's best for the person versus what's best scientifically, and it may not be black and white at all.
Awesome. Great discussion so far. There have been a few questions throughout the various chat features
Yeah.
about translation and FDA approval. So I'm going to ask one that I think captures a couple of those. So can our panelists talk about how this type of study design should be viewed in terms of translation into clinical care? Do you think this is something that should be done as a first step, then an RCT with less conditions? Or is there an already established hierarchy of how we might conduct studies and conduct analyses?
I'm going to let Noam take that one.
Well, if I'm not, I know there have been papers from clinicians who used an N-of-1 design just to treat their own patients. What I don't know is if that actually was the origination of the N-of-1 design. It might have been. So yes, this can be used clinically, but I tend to be thinking of other conditions rather than spinal cord injury. But if someone has high cholesterol, for some reason, even though there's a whole group of drugs called statins, some people will respond to one statin and not the other. And so you can, knowing that as their clinician, you can have them go through a series of phases. What is your LDL, which is the cholesterol level, on this drug, and then plan to change to another drug and change to another drug all in advance to see which drug works best for them. So it can be done clinically. For spinal cord injury, it's obviously longer term. None of us have quite moved the needle enough in terms of motor recovery or blood pressure stabilization, but obviously things are advancing and there's a lot of progress on that. I'm not sure how to answer that one.
So, so I'm not a clinician, so forgive me if I'm speaking at a turn here, but as a patient, it strikes me that every interaction with the physician is an N-of-1 type of experiment, right? So I come in and uh, I've got pulmonary hypertension, and so there's a variety of drugs you can try. There's doses you can try. You come in again, you see how things are. But this is what we're talking about here is a little bit more systematized, right? And we're trying to get, maybe I'll say, quasi generalizable results, right? What we're hoping is that a pattern emerges, like with the blood pressure. So Tommy knows the details better than I do, but there were just certain settings that just were not working for anybody, right? And we sort of quit looking at them after a while. And so that allows you to sort of focus on some other ones that seem to, qualitatively be more effective. And then we use that and iterate to the next step. Is that fair, Tommy?
Hmm. Anything else on that answer, or want to make sure I don't cut you off before I go to the next question?
Well, that's a great questions. Let's keep going.
All right. So we've had a couple of questions about funding. So I'm going to ask one question that kind of captures a few of them. So how does funding work for running trials with the small n, and what has your experience been getting funding? Because it's a struggle not just for spinal cord injury, but in any rare disease with small populations. So yeah, so can you just talk a little bit about that, about how it's been getting or not getting funding for studies with these types of designs?
Yeah, thank you for that. And actually, I saw some of those. I was hoping we'd get to that one. So this is anecdotal, but this is now 3, 4 years ago. I was getting a little frustrated. Those of you know me know I can get a little frustrated. And I was like, we can't get a proper RCT in our field, or at least not easily. Let's try something else. And actually, I kind of knew as I was writing these two grant applications, reviewers are either going to like it or they're going to just completely dismiss it. And we wrote two grants. We're doing something very similar to this in ALS, and it went two for two. That's all I can say anecdotally is two for two on grant applications explicitly saying we're going to use this type of design. But I think it's reviewer dependent, and as everyone knows, that's a box of chocolates. You don't know who you're going to get.
All right, so keeping on going down the line in the order that I'm seeing the questions. So this sounds like it might be a question more for you, Joe. The question is, I noted you said you don't necessarily aggregate at the group level. However, can you? Could you use Bayesian modeling to constantly update the combination to try and maximize efficacy for different injury typologies while still acknowledging heterogeneity? I'm thinking about how we deliver this type of design in a community facility with little control, trying to better prescribe a combination for a person with an SCI.
Good. Lots to unpack there. I guess I'm not sure what the question means by group level. So, um, we've got the subjects, and we have all these different settings that are sort of flowing out of that subject. In a more traditional study, those subjects are then part of the placebo group or the experimental group or something like that. But I guess if group means parameter settings, um, what seems to be the case is that averaging across, like let's say a given intensity level, and you've got 30 pressures that have a mean and a standard deviation, you could just take the average and use that as the score in your statistical analysis. Um, but I think there are some advantages in terms of like statistical power um, of keeping all those scores in there. Um, I don't know if that answers the question or not. Now, with respect to Bayesian, if I could start from scratch, I would probably be a Bayesian. But I've got like 30 years of frequentist thinking in my head, and it's really hard to kind of break some of that mindset. But my short answer is yes. One of my doc students, who's already a way better statistician than I am, is um, pretty pretty Bayesian in his approach to things, and there are advantages and disadvantages. I mean, really smart people argue both sides of frequentist versus Bayesian, but just from a philosophical perspective, I do think the Bayesian framework makes a lot of sense.
And obviously, not knowing the intricacies of the statistics, one important point to try to add on is the amount of data that you get from each participant in this type of design is more than you would normally get in an RCT. And we're absolutely going to need some sort of model based on data and machine learning slash AI to turn this into something that thinks for us in a community facility. We cannot expect in our healthcare system to do anything complicated or add more complications to community health. It's just going to be hard to implement.
Well, I will say though that you know, one of the reasons that we're taking the single case approach is that we don't actually have the same experimental conditions applied to each participant, right? In terms of uh, stimulus site, monophasic versus biphasic, all those types of things are not exactly the same for every participant because it is an adaptive approach. If you could, um, in a traditional RCT, if you cut down the number of parameters that you're looking at, you can fit all of this into a more traditional experimental design and also keep every one of those data points. Um, so whether it's single case design or a regular RCT, if I've got 30 pressures at a given stimulus intensity, I want to have all 30 of those scores in my data frame.
All right. So I want to preemptively let everyone know one of our speakers does have a hard cutoff at 1 p.m. So we've got time for one more question. Hopefully he doesn't have to leave in the middle of it. But if we didn't get to your question, we'll be saving the questions from the chat and we can follow up via email. So I
There are a lot of great
want to
questions.
thank both of you for awesome presentations before we get to this last question. Um,
There are a
so.
lot of great questions. We'll get to all of them, whether or not it's live.
So the last live one, can either one of you go into more detail about designs like ABA or any other designs that might be relevant to dense physiological data like blood pressure?
Yeah, maybe I'll quick comment on that. And also, I do have to leave too. I've some students who have to take a makeup exam here that are outside my door. So yeah, that ABA design is kind of traditional old school single case, as I understand how it's been done, like in the social sciences forever. Um, so you could have, let's say something simple like an antihypertensive med versus a placebo, right? You can take serial blood pressures with drug A or pill A, and then with appropriate washout, you could take serial blood pressures with pill B, and you could flip back and forth like Noam showed on one of the earlier slides. And you could have hundreds of data points for pill A and hundreds of data points for pill B and analyze that data. Um, you still have like, maybe order effects, right? So A1, B1, A2, B2, some of those types of things would still need to be, I think, considered. But yeah, this is all doable in that sort of ABA framework. Now, what we're doing here is not an ABA framework, right? Um, so, um, and as a little bit of background, when Noam first contacted me about doing this, he sent me, it was a linguistics paper or a speech paper. And I still have it. It's not an easy read. So people have been doing this type of stuff in the life sciences for quite a while. But it's not, it's not that straightforward to pull off, as I'm unfortunately finding out.
You mean everything is harder than it looks?
Well, now I got a really good grad student, so I just give it to him.
All right. Well, with that, again, to respect both your time, thank you both very much for an awesome presentation. And again, we'll be following up with questions that we didn't get to live in email, so stay tuned. And we'll be continuing this non-traditional research design seminar series through the Asia Early Career Committee this summer. So stay tuned for more information about that. And everyone, have a great Friday and great weekend.
Thank you, guys. Thank you very much.
Thank you so much.
Great talk.